Hey, good morning, everyone. So we left off last time talking about um, allergic responses, and one of the ideas is that allergy is a misdirected response that would normally be targeting parasitic infections, including helminth or hookworm infections. And because we live in a cleaner environment and typically don't get helminth infections, instead the immune system is attacking environmental antigens. And so one of the approaches to this, sort of, which is not a medically approved approach, but you can do this, is to buy hookworms, ingest them, and have them infect you. And the idea is that then your immune system would start targeting these instead and reduce allergies. Now, the question is, well, tell me why this would or would not be a reasonable therapy. And I'll say that I would come down on the side of this is not a reasonable therapy, even though it may redirect the immune system uh, against helminths. It still has other problems. The first of which is now you're infected with hookworm, and that can have serious complications, including uh, problems with the digestive system. From an immunologic basis, though, the problem is that once you've already started an immune response to something, for example, to pollens, the just making an additional response to hookworm is not really going to help you. Instead, a more conventional approach to resolving allergies is to desensitize with antigen, in particular, getting a lot of regulatory T cells to suppress the response that's already there. Okay, so we've covered sort of immune responses we want to foreign things, and then immune responses we don't want to self or foreign things. And now we're going to talk about immune responses that we do want to self. And this is sort of autoimmunity that we want. And that happens uh, when you get cancer and you want your immune system to attack it. The current estimates are that you develop at least one cancer cell every day of your life, and the immune system controls it until the one day that it doesn't, and the cancer progresses. And so there's a lot of evidence here for what the immune system does during cancer. And the idea that this was happening goes all the way back to Ehrlich, right? Ehrlich is really the grandfather of immunology, where he showed that you could get responses to foreign things. He proposed the idea of autoimmunity, and he was really against that. But then he said, well, to sort of uh, minimize that opposition, he said, well, maybe the immune system can recognize and eliminate tumors, and so that would be a self-response that he was in favor of. Now, there really was no evidence of this, of the tumor surveillance or the idea that the immune system could function to resolve tumors. But if you remember, in the mid 50s, we had the idea of the clonal selection theory, one of the people being McFarland Burnett proposing this. And these were theoretical immunologists that really didn't have um, any knowledge of what the immune system was doing. But they did know from Ilya Mechnikov and others that cellular immunity existed. And so they were trying to reconcile both humoral immunity, the presence of antibodies, and cellular immunity. And they were saying that, for example, Lewis Thomas in the mid 50s said that the main the main duty or the main function of cellular immunity is not to promote allograft rejection, because why would you evolve something to attack transplanted organs? That doesn't make any sense. But instead, cellular immunity that they knew would attack transplants was really designed to protect you from cancer or neoplastic disease. And so McFarland Burnett, who developed the clonal selection theory, said, well, okay, maybe tumors develop uh, different antigenic properties from self cells, and that allows them to be recognized and limited by the immune system, and so therefore you don't get disease. Now, this is all hypothetical. There was no proof. And the first proof was actually negative. So Osias Stutman in the mid 70s started to test this. And the hypothesis is that, well, if the immune system is designed to, to control tumors, then if somebody is immune suppressed, then you should have higher incidences of tumors compared to normal people or to people with intact immune systems. But the problem was when you actually looked at humans who were immunosuppressed, there was really minimal risk for developing cancer. They had no increase in sort of uh, spontaneously occurring colon, breast, lung, prostate cancers. Those didn't really support that. There was some slightly increased rates in immune compromised individuals where there's a pathogen that led to cancer. So H. pylori can lead to gastric cancers, papillomavirus can lead to warts, herpes simplex virus are associated with some cancers. But that implies that you're surveying for the pathogens, not for cancer. So what Osias Stutman did was he treated mice with 3-methylcholanthrin, which is a DNA intercalating agent, and it will cause mutations and lead to the appearance of cancer. And so he took nude mice, which don't have a thymus, and so they don't have T cells, and treated them with this compared to normal mice, and there was no difference in cancer incidence or progression. That suggested, no, the immune system is not doing this. That was really where it stood for a while until Bob Schreiber at Washington University in St. Louis sort of repeated the experiment, but instead of using nude mice, he used RAG2 deficient or interferon gamma receptor deficient or STAT1 deficient mice. And what he showed is they did indeed have increased incidence of cancer and increased progression. So you can see that on the left graph, RAG2 deficient mice, more of them developed tumors and the tumors progressed faster. And so on the right, you can see, well, they looked at RAG2 deficient, interferon gamma receptor, STAT1 deficient, or RAG and STAT1 deficient. Now remember, STAT1 is downstream of the interferon gamma receptor, so you expect those to be the same phenotype. What he found is if you knock out both, you actually get even higher incidence of cancer. So this was the first proof that the immune system does something to control cancers. And it was related to development of lymphocytes because RAG2 is only expressed in lymphocytes. And it was related to Th1 type responses where interferon gamma is important. So the question is, if that's what's happening, how is the immune system doing this? The first thing that we need to keep in mind is that tumors are not normally vascularized structures, okay? When a cell becomes transformed, it often becomes hypoxic because it doesn't have appropriate oxygen flow. And so when that happens, the hypoxic environment actually results in increased expression of ligands for activating receptors on NK cells and as well as macrophages and other dendritic cells. And so what's happening here is you get activation of NK and NKT cells. Now it turns out the ones that are most important here are the NKT cells because they sort of get everything going. They're like the spark that lights the fire. And so this explains why RAG deficient mice don't initiate an immune response against cancer, but nude mice do because NKT cells can develop outside of the thymus. Okay, so NKT cells will get activated by dendritic cells or in some cases macrophages that are sitting in that tumor environment and they're producing interferon gamma. Interferon gamma is going to then signal through the interferon gamma receptor and STAT1 to activate NK cells, and they further get the ball rolling by starting to kill transformed cells. That then leads to something similar to what we saw in autoimmunity of epitope spreading. Well, now you have antigen being released from the killed cells, and so you can have antigens presenting cells taking that up and initiating a T cell response. And so we know that tumor control, the vast majority of tumors require CD8 T cells because those are the main cells that are going to respond and kill tumors. So you have to get innate triggering first, that's through the NKT cells. Then you get NK cell activation and killing of tumors, and then the dendritic cells get activated and take up those things and then are able to present them to CD8 T cells. And that's the gist of what we're going to talk about today. But there's some problems with that. The first is, what is the innate triggering? Well, we know the hypoxic environment can do this, but then the dendritic cells need some activation signal. And typically we think of dendritic cells getting activated by PAMP receptor, pathogen-associated molecular patterns. But this is self. So what are the self PAMPs? And this was proposed by Polly Matzinger, who's a very interesting figure in immunology. She once put her dog as a co-author on a paper. And she proposed what was at the time called the danger hypothesis, that it doesn't that danger or a signal for the dendritic cells doesn't necessarily have to come from a pathogen. It could come from self-proteins. And at the time, she called those alarmins because this was prior to the recognition of toll-like receptors and definition of what all of our PAMP receptors are. But they're really things that are released from a cell that shouldn't normally be released. So it could be things like DNA or DNA packed around histones, mitochondrial proteins. It could be high levels of extracellular ATP. And those now we call damage-associated molecular patterns, or DAMPs. So PAMPs are from foreign things, but DAMPs are from your cells that get killed. Okay, so your NK cells that get activated by interferon from NKT cells are going to initially kill some tumor cells. Those then are releasing the DAMPs. That then the dendritic cells are picking up the proteins from that cell as well as getting activation signals from the DAMPs. So this brings us back to, why is there a difference between nude mice or athymic mice and the RAG2 deficient mice, or the other ones that Bob Schreiber looked at? Well, the first thing is RAG2 deficient mice don't have NKT cells. Nude mice do, because they can develop outside of the thymus, and so there's a difference right in the beginning of the spark that could light the fire. Okay, so the NKT cells are required to provide activation to the NK cells to get some tumor killing that really starts this process. If you don't have those, then you don't get NK cell activation. You get a reduced ability to initiate responses to tumor, and that then leads to a reduced B and T cell responses. Because of that, then the tumor can grow. Now, if you look here, you can actually see there are a number of times when you have things like interferons are being produced, type interferon gamma. So here our NKT cell is getting activated. It is producing cytokines that then can activate other cells. For example, NK cells can also activate or help bias the T cell response to a Th1 response, as well as activate CD8 T cells. And those are all able then to go and kill the tumor. In addition, we remember interferon gamma is going to bias your T helper response to Th1, and that will bias it towards production of IgG. IgG then can bind to tumor cells and be recognized by FC receptors on the NK cells and get further killing. So B cells do play an additional role here. But it really comes back to, you need this initial guy that can come in and be activated and activate all the other cells. So you get the initial spark. So what happens is then your NKT cells get activated. And it's not entirely clear still if this is through dendritic cells or direct recognition of tumor cells. I think there's evidence for both. But in reality, the NK T cells are being activated, and they're going to make interferon gamma. We haven't really talked much in this course about gamma delta T cells. They essentially can do the same thing, but they have a more limited distribution to mucosal epithelia. Once those cells make interferon gamma, they're first going to give you NK killing and release of antigens. That then leads to these a little bit out of order. Then dendritic cells will take up those antigens, migrate to a lymph node, and activate CD8 and CD4 T cells. And those then come back to the tumor and start killing it. Remember, a Th1 CD4 cell is really a wannabe CTL. It can kill not as well as a CD8 cell, but it can do a lot of those same things. Okay, so that's how you initiate a response to a tumor. The second question is, what are the T cells seeing? Because remember from our autoimmunity lecture and talk uh, about tolerance, we have both central tolerance, which should delete most of the self-reactive cells, and peripheral tolerance, which should shut this down. And so you have to overcome those things. Part of that is, well, what are they going to recognize? So there's two terms that are often used, and they're used primarily by oncologists who are perhaps the most adept medical immunologists. And the terms are tumor-associated antigen and tumor-specific antigen. Now, the more common of the two is tumor-associated antigen. And so it can be present on tumors and on some normal tissues. Okay, and so many of these end up being fetal antigens that tumor will express, but they're only expressed during developmental stages. And so in an adult with cancer, that's a tumor-associated antigen. It can also be that the tumor has a lot more of that than normal tissue, so the immune system is biased towards attacking cancer cells rather than self. But that often crosses over. Okay, so these are usually overexpressed or improperly expressed self-antigens that are expressed at specific stages of differentiation for the normal cell, and when you get a tumor, it dysregulates that, and so it opens up the potential for seeing something. The more rare version of this is a tumor-specific antigen. This is an antigen that's only on the tumor cell and is not expressed anywhere else. And so there are, of course, if there's a virus associated with a tumor, then you can have, those are only going to be in tumor cells. They're not going to be in normal cells. It can also be, as I said, proteins that are not expressed anywhere else in the body, that are only expressed in fetal stages or in cancer state in cells. And so if you have a protein like that, it's not expressed anywhere else at the time you have cancer. It can be also, particularly for B cell antibodies against cancers, it can be altered glycoproteins or glycolipids that there is a mutation that leads to alterations in how you glycosylate something. And that doesn't occur anywhere else in the body. Or it can just be simply mutations that lead to epitopes that are not present anywhere else. There are also some examples. Hootie Warren at the Fred Hutchinson Cancer Center has shown that in some cancer cells, you can actually get post-translational splicing of proteins that then can be recognized by the immune system. So those only occur in tumor cells.
I had a quick question.
Yes.
Do we know like what percentage of memory cells in your body are tumor specific versus pathogen related?
It depends, but it's still very low. You can detect them, and we'll come back to that when we talk about immunotherapy, which is stimulating those cells, but it's can be below the level of detection up to maybe 1%. But I'll come back to that because we don't actually know a lot of antigens. But I'll sort of come back to your question. If I don't, please remind me.
Thanks.
Okay, so how do you get these T cell responses? Well, you remember to stimulate a T cell response, you need dendritic cells. And so initially, you get NKT cells, activate NK cells, and NK cells will kill the tumor. When they do that, they'll take then the DAMPs that are released will activate the dendritic cells. They'll also be at the time taking up parts of the necrotic tumor cells, and they'll be presenting that to T cells. Okay, so for the class 2 pathway, this is the normal endocytosis and degradation, resulting in loading of peptides on MHC2 in the M2C compartment. For MHC1, though, remember this is an extracellular thing that's being taken up, so you really need cross-presentation. That's where you're going to start to digest this in the endocytic pathway. It feeds through that Sec61 pathway into the cytoplasm, is pushed through the immunoproteasome, and then back out through TAP and loaded onto class 1. Okay, so this is why the CD103 positive dendritic cells that do cross-presentation are so important, particularly for tumor responses. Once that happens, though, they'll have DAMP signals from the necrotic tumors. They'll start taking up antigens, and so they have both of the things that they need. They'll get informed to go to a lymph node, and they'll stimulate your CD8 and CD4 T cells. But it has to be to these tumor-associated or tumor-specific antigens. Once that happens, then you'll get CTL and you'll get Th1-like helper responses that then can go out and start to control the tumor. But the problem is that it's still difficult to identify tumor antigens. And so the first way that this was done was something called a Serix assay. This is a serum-based expression library. And the way that this works is that first you have to make the assumption that if you have an antibody responses to a tumor antigen, you are also likely making T cell responses to epitopes derived from that antigen. Okay, so there's an assumption there. It doesn't necessarily mean that you'll have T cell responses, but it just enriches for them. So what do? You have a patient, you take the tumor and you isolate all of the mRNA from that tumor, and then you convert it to cDNA and then put it into a phage, and you just make a library of all the different genes that are being expressed. Okay, and then you're going to take that library of phage and infect E. coli. Okay, so it's going to now express the RNA, or sorry, it's going to express the genes that were being expressed in the tumor. Now on the other side, you're going to take serum from the same patient, and you're going to add it on top of that infected E. coli lawn and see which antibodies, or you're seeing where are the antibodies binding. Okay, so in this case, the antibody has beta galactosidase. So wherever there's antibody bound, the plaque looks blue. So then all you have to do is you isolate the phage from that plaque, you go back in sequence and say, oh, okay, it was recognizing this gene. Okay. Now, this doesn't guarantee that there will be a T cell response, but it does narrow down the list. And people like Lloyd Old, who we've talked about before, spent their entire scientific career trying to identify T cell responses. Well, this worked okay, but it was really not a terribly efficient way of identifying what are the T cells seeing.
Dr. Blackman,
Yes.
is that assay similar to like an ELISA spot?
The readout is, but then what you have, an ELISpot is just giving you dots that you count. But in this case, you're looking for antibodies. So the antibodies are what is causing the infected E. coli to turn blue. And so then you're going to pick that colony, or that, I guess it technically is pick that plaque, and resequence what the phage was expressing. So you're going all the way back to here, saying what in that phage was the antibodies recognizing. So what you had to do then is confirm that what the B cells were seeing was also being seen by the T cells. And so what they would do then is take the cDNA that was identified from that Sirix method and put it into some mammalian cell line, or clone those genes into a mammalian cell line, and then incubate it with T cells from the patient. Okay, and most of the time, no, the T cells weren't seeing that same antigen. Okay, but sometimes the gene that you identified from the Sirix method would lead to killing of the target cells, and then you would have a positive response. Then you knew, yes, in fact, the T cells could see that. Now, the problem with this is, if you're going to do this in humans, you have to have both the T cells and an antigen presenting cell, or some cell that can you can express this and get the antigens loaded on the class 1. Well, we're an outbred population for the most part, and so you have to have MHC matched cells each time you do this. Okay, so that really makes it harder. It's easy in mice or smaller rodents where we have established cell lines. They all have the same MHC. But what you have to do then is basically be able to grow the T cells. And this was enabled by Kendall Smith and others who identified T cell growth factor, allowed us to keep cells alive. But now it's a little harder. You have to have MHC matched target cells. The way this is typically done is to take PBMC from a patient and transform those cells with Epstein-Barr virus, and so you have an immortalized cell line. But then that leads to alterations, and most of the time you're seeing just Epstein-Barr virus epitopes get expressed, and so it's not a great way to do it. But from that, we've identified a number of tumor-associated antigens. And as I said, Lloyd Old really led this effort for a long time. There's some more targeted ones are Wilms tumor antigen. You can see WT1 is expressed in a number of cancer types. There are things like the melanoma antigens. Okay, so these are PRAME antigens, PR1. You can see those also occur in a number of different antigens. There's some that are sort of specific. You have prostate specific antigen. You can have NY-ESO-1, which is in myelomas. You don't have to memorize this table. Just know that there's actually only a handful of identified, confirmed tumor-associated target antigens. Okay. Now, when you talk about these, then you have prostate cancer has a prostate-specific antigen. So you don't want to necessarily wipe out the rest of the prostate. Okay. So you have to then talk about what are acceptable response or acceptable losses. For example, testicular cancer, typically if your immune system is going to respond or you're going to target something, you're losing the testes. This also happens in ovarian cancer or breast cancer, where we're talking about tissues that you don't want to lose, but you'd rather stay alive. And so that often is an issue when you're talking about tumor-associated antigens is, well, what's going to happen to the rest of the normal tissue? Now, what happens then is that the T cells have to get to a tumor, okay? And the more T cells that you get in a tumor, the better off your response rates are going to be. But the problem is that tumors are not vascularized, and so they're often very solid, and it's very difficult for T cells to get in there, and that then makes them inaccessible. So you really have to have this ongoing battle between hypoxia, which is going to start killing the tumor cells and releasing antigens. That's great. But then if you get a response, it's not great because then the T cells can't get into it. So sort of these come in waves, and the idea is that we want to increase T cell infiltration and retain hypoxia. Ultimately, as tumors progress, they will start to become vascularized, and then the response can get to it. But we do know that the more T cells you get into a cancer, the better off you are. This, I forget which cancer, I believe this was melanoma. But basically, you have the T cells are responding to tumor antigens. And if you segregate patients to those that have high T cell infiltrate, so the H here's lots of T cells, you can see that they survive better. And if you have low T cells, that they have a worse prognosis. Okay, so more response is more better. But that doesn't always work. Okay, immune escape can and does happen. For example, these patients all had good T cell infiltrates, but they still died. Okay, so it's better, but it's not always going to work. And why is that? Well, the main thing is that tumors are good at evading the immune system. We talked about how viruses can do it. Tumors essentially use a lot of the same tricks that viruses will use. A infected cell, the virus doesn't want to be killed, and so it will start evading the immune system. Well, tumors, if we anthropomorphize them, they don't want to be killed either, and so they're going to evade the immune system. In reality, it's an evolutionary process where they're making random mutations, and then the ones that survive are the ones that have done something to evade. So we go back to Bob Schreiber's work. And so he did provide the first evidence for immune control of cancers, saying that yes, you need RAG proteins, you need interferon gamma and interferon gamma receptor and STAT1. We need all of those things. But what he also showed, which was perhaps more important, was that tumors that have undergone that have eventually evolved, even though there was an immune response to it, are actually more aggressive. And that led to what's called the immunoediting hypothesis. If you have, here's our tumor, okay? If we have a great T cell response, right, we get our NK T cells, those activate NK cells, then you get dendritic cells that activate CD8 and CD4 cells, get a great response, you can control the tumor. But the problem is, say we had a tumor, we got some activation, it wasn't quite enough to really control the tumor, and now the tumor is going to start to mutate. So you get all these different colored tumor cells here. And that then leads to a constant battle. The tumor is being controlled by the immune system. You select for tumors that are resistant, that results in other problems. And so this can go on for quite a long time. This equilibrium phase where the immune system is battling the cancer. And it continues to, with each change, adapt to that and then control it. Now, for some cancers, for example, melanoma or some things like pancreatic cancer, this can be many years. Some of our estimates are that when you finally show up in the clinic with an actual tumor, that tumor has been in your body for 20 to 30 years. So it can be quite a long time. What happens then? Why do you show up at the clinic? Because eventually this tumor has figured out a way to escape from the immune system. So it can suppress the T cells. You get lots of where the immune system is designed to shut down. So you're going to get regulatory cells that are going to come in and they're going to block all of those immune cells. And when that happens, now the tumor's got a free reign. It can go ahead and proliferate and the immune system can't do anything about it. And this is the idea of why immune checkpoint blockade, where you're using anti-PD-L1 or anti-CTLA4 antibodies, can work because the immune system's there, it's just being suppressed. So that is the basis of immunotherapy. Okay, so eventually this equilibrium phase is going to keep going on until the cancer wins. And then when it wins, then you're going to have an actual tumor. So this is what Bob did, okay? And the first thing is that, okay, he took mice, he treated them with 3-methylcalanthine, that's the tumor-inducing agent, and he separated them. There was the mice that did not show any tumors or had minimal tumors, and the mice that progressed, okay? So you can see here, these mice progressed. Okay, so the immune system was not able to control them. But these mice had minimal tumors. Okay, so they're down here. And what he did was he showed that the immune system's actually doing something there. And so he treated those mice with anti-CD4 or anti-CD8 antibodies or anti-interferon gamma antibodies. And you see, as soon as he did that, those mice all started progressing. That suggests that the T cell responses and tumor are existing in equilibrium, and neither is dominant. Sort of the immune system is controlling it. You can see if you don't treat with anything, eventually they're able to control it. But if you do block the T cells, then the tumor progresses. Okay, so that's the first evidence that the immune system is actively trying to do something, even even in the mice that don't have much of a tumor. The second thing he did was to take tumors from mice that either did have an immune system or did not. And so if you take, so first of all, here's the starting tumor, okay? So the starting tumor, basically if you put it in, sorry, I explained that wrong. There's a lot here that you could look at, but let's start with the tumors from RAG deficient mice. And you can see here that if you put those in, about 40% of mice will get tumors, suggesting that's okay, that's normal. So the real comparison then is this is a mouse that is more susceptible to tumors. This is a mouse that has an intact immune system. Okay, so he isolated tumors from those two groups and compared them. And the only two I want you to focus on are the blue lines and the green lines here. If you take the tumors from a mouse lacking an immune system, you get some growth, but if you take tumors from mice that have had an immune response to the tumor, you can see that they progress much faster. And what that suggests is that there is selection for an immune resistant cancer. Okay, so that really was the first proof that there is a response during cancers that's maintaining it, and that after that fails and you get a tumor, that tumor is now immune resistant. How does this work? What is tumor escape? Well, most of the time, on the left we have sort of our normal scenario, right? We've had an NK cell, it's activated, or an NKT cell has activated an NK cell. You get some tumor killing, processing and presentation of antigens, and activation of CD4 cells and CD8 cells, and then they come back and they kill the tumor and eliminate it. That's fine. That's what you want, and that's what happens most of the time. But there are problems with that. First of all, if your tumor is going to escape, most of the time what it's doing is using your own tolerance mechanisms to shut down that response. We know that T cells are designed to shut down, right? Because if they didn't, they're going to kill you. But also, you have your normal tolerance mechanisms. It can be regulatory T cells. We know they can bind all the IL-2 and sort of keep the other cells from dividing, but they make suppressive cytokines like tumor growth factor beta. That is how it was discovered. It was a cytokine being made in a tumor. And if you express it, tumors would keep going. And then what it's doing is shutting off the T cells. You can also have, if you have an immature dendritic cell, right, it didn't get those damp signals. It presents antigen to a T cell, well, hasn't upregulated co-stimulatory proteins. And so that blocks the T cell response from happening. Okay. More recently, there were some myeloid-derived suppressor cells. Those have a role in tumor suppression of the immune system. And some tumor cells themselves can develop these properties by mutating. So those all happen within what we call the tumor microenvironment, where you're shutting down the T cell response. But there can also be other things that the tumor is doing. And this is a really interesting pathway that is normally, that tumors are normally doing to block the T cells from getting into a tumor. What tumors will do is they will express high levels of the endothelin 1 protein, or ET1. That it then will bind to the ET beta receptor or endothelial B receptor. And what that does is then that causes block of expression of ICAM1. Well, remember, how do T cells get into tissues? They're getting activated by a chemokine that activates the LFA1 protein. LFA1 recognizes ICAM1. So if you just downregulate ICAM1, the T cells will keep on going by, and they won't be able to enter the tumor. Now, the flip side of this is really interesting. And they also are making the ET1 that that will activate the ETA receptor or endothelin A receptor on tumor cells, and that will cause them to express metalloproteases. What does it do? Well, those are causing tissue remodeling. Extracellular proteases are digesting connective tissues. It gives the tumor room to grow, but it will also express then vascular endothelial growth factor. That's going to recruit cells that are going to make blood vessels in the tumor. So now the tumor can spread throughout the body. So that's the metastasis of it. Okay, so a tumor by expressing ET1 is both blocking T cells from getting the tumor and setting it up for being able to grow within that site as well as to migrate to other sites. But it's often the escape from the immune system is often much simpler, right? So we know viruses are very good at mutating. HIV-1, for example, which we'll talk about in a subsequent lecture, just mutates the epitope, and so now you no longer present that to a T cell. So even if you initially had a T cell response to a tumor antigen, some epitope from a tumor, and then you start killing off that tumor's mutating, and it then mutates that epitope. Now the T cells can't see it. Okay. And so it can either be you mutate it or they just don't make that protein anymore. And so could be either of those two scenarios, and they both happen. But this is why we have such a limited number of tumor-associated antigens, because essentially for you to identify those, you have to look at antigens that are necessary for tumor survival. If they're not necessary for tumor survival, tumor will just mutate them, and then you essentially don't have any T cells that can recognize the tumor. The more common thing that tumors do will just stop expressing MHC proteins. Okay, so there's our tumor cell. Initially, you get a T cell response, and it starts killing off those tumor cells. But then the tumor will just say, okay, well, I'm not going to make any MHC. Then the CD8 T cells can't see it. Now, this does make the tumor more prone to NK cell recognition, right? Because NK cells are getting that don't kill signal from MHC. If you lose that, then the NK cells can kill them. But the problem is that NK cells are really designed to initiate responses. They're not designed to be the end-all controllers of the tumor. They're just there to help initiate the response. And so the key here for the tumor is to escape from T cells. Even if they're a little more susceptible to NK cells, they're still escaping the majority of the response. So that's great for the tumor. The other things that it can do then is to, of course, what we talked about is inactivate T cells. So tumor cells, particularly if they spread to a lymph node, they'll present antigen to T cells in the lymph node. Right? So this is often where you hear about when a tumor metastasizes, it's using your lymph nodes to move around. Right? It's going through the lymphatics to a lymph node and eventually back into the blood. But when it gets to the lymph node, tumor cells don't typically express costimulatory proteins. And in fact, so here it's just not expressing the costimulatory protein, but in fact, what it also can do is start expressing inhibitory proteins. And so that leads to those things that we talked about, either anergy or death of the T cells that can recognize the tumor. So it's nipping the response in the bud, where it's before the T cells get going, you have the cancer cell is stopping the T cell response. But even if you have a T cell response, right, you You made a response to your tumor. The T cells are not designed to keep going. And particularly for CD8 T cells, they're designed to shut off because if they don't, they're going to kill you. So there are a number of inhibitory proteins that T cells are going to make. And so some of the more important ones are PD-1, there's TIM3, LAG3. There's a number of other ones that are expressed, and those are inhibitory proteins. And their main job, what most of them do, is that they bring in phosphatases to the contact region between the T cell and the tumor cell. And those phosphatases are preventing the kinase cascades that you need for T cell killing. So if you prevent that, you basically are saying, no, you cannot kill this tumor cell anymore. This is perhaps the most common pathway, and for all of the immune checkpoint blockade approaches, they're really targeting these interactions between the inhibitory receptors on the T cell and the inhibitory protein ligands on the tumor cell. So one of some of the main ones, PDL1, PDL2, there's an anti-CTLA4 antibody. Those are all designed to block these inhibitory pathways and allow the T cells to actually do their job. And finally, or I don't know if this is finally, we have sort of this idea in the tumor environment, you really have regulatory cells in particular are making this protein called tumor growth factor beta. This is normally functions in peripheral tolerance to shut down autoimmune responses. But you can think of a cancer response as sort of an autoimmune response. And so you get expression of TGF beta. And that will block CD8 T cells, block CD4 cells, can inhibit NK cells, as well as shut down dendritic cells. Okay, so it's blocking responses that are already there, but it's also preventing new responses. Okay, and it does that in a number of ways. You don't have to memorize all these ways. Just know that TGF beta is suppressing other cells. And so if you get a lot of cells making that, everything else falls apart. And so we come back to these regulatory cells, right? These are really important for maintaining tolerance to self and preventing autoimmunity. But if a tumor is derived from you, it looks a whole lot like a normal tissue. And so these pathways become dominant. Now, it can be either natural Tregs, which are the ones that come directly out of the thymus, or it can be an induced Treg, a normal T cell that responds but gets inappropriate signals or gets suppressive signals and shuts down, or becomes a regulatory cell to shut down other cells. There's a number of ways that this happens. It can be T cells that are activated on tolerogenic dendritic cells. It can be on tumor cells themselves. And it can be the influence of these myeloid-derived suppressor cells. But essentially, there are a number of different cells that can give you these. And it basically creates an environment in the tumor where the immune system just can't work because you have all these these tolerance mechanisms that normally would prevent autoimmunity are preventing an autoimmune response to the tumor. So what do you do about it? Right? What is the approach to cancer? Well, by and large, chemotherapy has been an immense failure. It does work in some pediatric cancers pretty well, but in adult cancers, chemotherapy can reduce the cancer, but eventually it's going to come back and kill you. The modern idea of treating cancers is to harness the power of the immune system to eliminate it. Okay. And so what we do know is that if you have cancer and you don't treat it, you have a certain normalized survival curve for that. Chemotherapy can push that curve out, but it typically doesn't cure most patients. It's just basically giving them extra time to live. On the other hand, if you have immune modulation, you can push that curve out, but you can actually cure many more patients. Right? So you're harnessing the power of the immune system. And as we get better at doing that, it's going to improve survival rates overall. So what do we do? Well, you remember there's this endothelin 1 pathway, right? That's blocking T cells getting into the cancer and also promoting cancer remodeling and metastases. Well, there is a drug called BQ788. I forget which manufacturer makes this, but it's basically binding to the endothelin B receptor and blocks it. And so you don't downregulate ICAM1. The T cells are able to recognize that and get into tumors and kill them. So you're basically recruiting more cells to the tumor. Because we know the more cells in a tumor, the better prognosis you have. So the first thing is get them there. The second thing is to promote an environment which you're going to have lots of killing of the tumor and inflammation. And this is something that is not really done. There's really only one group in France that does this anymore. But the idea is you put in pro-inflammatory cytokines into the tumor. Now you can really only do this with isolated tumors. So it has to be in a peripheral, in a leg or an arm, somewhere where you can actually blockade everything off. And the reason for that is that most of the time what you're using is the most dangerous protein in your body called tumor necrosis factor. It's great at killing off tumors, but it also does a lot of other things, including can send you into hypoxic or into shock by having your blood vessels leak. And so what you would do is then, if you have a tumor here, you then put a tourniquet around that organ, around your leg, and then you're going to infuse TNF directly into the tumor. Now this works really well, okay? It can actually cause tumor regression. The problem is that this is a very dangerous thing. If that tourniquet is not applied correctly, TNF injected into the rest of your bloodstream will essentially cause you to bleed out internally and die. Okay, so it's a very aggressive approach, but it has its dangerous side. Now there are other variations of this, right? The first one that was ever used was actually something called Coley's toxin. This was just basically heat-killed bacteria that were injected into a tumor, and those then led to, in some cases, tumor regression. What is it doing? Well, essentially, you're providing DAMP signals, right? Or, sorry, in that case, you're providing PAMP signals, pathogen-associated molecular patterns, that then gets everything going. You can use oncolytic viruses. That is one approach that's being developed at ASU, is to use viruses that specifically infect tumor cells and cause them to die. You can use localized radiation, so you get more DAMPs released, or you can use other cytokines that are not going to necessarily kill you, but basically generate more inflammation. All of these are really trying to do is generate more DAMPs, kill some of the tumor so you get more presentation to T cells, sort of get the ball rolling again. Now there's some more developed immune therapies. One of those is antibody therapy. Now this works well if you have a cancer that is either bloodborne cancers, so leukemias, myelomas, things that if you were to inject an antibody, it could actually bind to a tumor. It doesn't work well for solid tumors because the antibodies don't really get into the tumor, and so there's not really efficacious because of that. And there's some variations of this. You can just use regular antibodies, okay? And then those bind the tumor. You have NK cells or macrophages will come in, recognize the FC portion of the antibody, and then kill it. So that's antibody-dependent cellular cytotoxicity. That's essentially an autoimmune response that you're trying to target a cancer rather than, a normal organ. Some variations on this are you can attach toxins to it. So the cancer cell will, expressing that antigen, is bound by the antibody, and then the cancer cell is actually internalizing that, and the toxin is killing the tumor cell from the inside out. This has a few examples that have worked, but it's very difficult because if that antigen is on any other cell except a tumor cell, you're going to start killing a lot of normal cells. Another variation of this is to put a radionuclide or isotope onto the antibody. And so when it binds to that tumor, then it's causing lots of mutations and the tumor will undergo cell death. The problem with that is tumors are already mutating, and so this hasn't terribly worked out well. Again, it's difficult to inject an antibody with an isotope attached to it and not affect other organs. The antibody is going to be cleared out in the kidneys, and now you've got a radioactive kidney that you're potentially inducing more tumors. So it's not a really great approach. The better approach is to use the cells that we know are important for controlling cancer. And those are really the T cells, and in particular, CD8 T cells. And so what Phil Greenberg and Steve Rosenberg have done, different variations of this, is you take a tumor and you isolate the T cells. And you're making the assumption here, well, the T cells in the tumor, it's probably responding to a tumor antigen. That's a pretty good assumption because T cells just don't migrate to tumors willy-nilly. They have to be, they're there because they are recognizing the tumor. You then take those T cells, either clone them up, so you select one cell per well, and then grow them, sort of the Phil Greenberg approach, where you're using clones of T cells. Or if you're Steve Rosenberg, you just take all those, grow up all the T cells, skip that cloning step, grow up all the T cells, and then infuse them back into the patient. Now, this has been facilitated by our ability to use the discovery of T cell growth factor and other cytokines that can promote long-lived T cells. Essentially what you're doing then is you're injecting them back into the patient. Instead of the one cell that you got here, you're injecting billions of cells. And so you have much more tumor reactive cells that can go and recognize the tumor and kill it. One modern variation of this is that you irradiate the host before you put them back in. And what that does is that it creates space. So if the T cells what they'll do is they'll recognize there's no other T cells here, I'll undergo more divisions. So you put in 1 billion, you actually get out 100 billion after in a irradiated patient versus a normal patient. So this is from Steve Rosenberg's group in New York. They put in these T cells and they could find these melanoma reactive T cells in the patient, and they were up to 75% of all the T cells in that patient. So coming back to the question of how high is the frequency, can be in patients undergoing immunotherapy, it can be quite high. And then if you look, if you look at the other T cells in that patient, they're just a very small percent. Okay, but we know this works because this is, apologize for that, excuse me. If you look at either tumor-free survival or overall survival, you can see that if you give this chemoablation prior to T cell infusion, that you can really increase tumor-free survival or overall survival in those patients. Okay, so this is some trick that we actually know works. There's some other versions of this. You provide IL-2 or other cytokines to the patient, and that helps those T cells survive. So more cells is more killing. That's what we want. And the better you can help those cells survive, the better off you're going to be. Now, many of you probably have heard of CAR T cells. And this is really just a variation of that same thing, but instead, what you're doing is reprogramming the T cells to recognize something they wouldn't normally recognize. Okay, so the one version of this is you take T cells, you isolate the tumor reactive T cell receptors, and you put those into some other T cell. Okay, so now this T cell is essentially expressing its normal receptor and the one that you put in. And then now you can grow those up and put those into a patient. This hasn't worked so well, and it's actually very laborious because you have to redefine what that T cell receptor is for each and every patient. right? Because they have different MHC. T cell receptors are not necessarily going to be the same. Antigens are going to be different. So it's, yeah, you could do that. And if you had enough of those on the shelf, yeah, you could just take one off and say, this is what we think is going to work, and put it into another T cell. The benefit of that approach is you can actually use T cells that populations of T cells that inherently have long-lived survival, so you can genetically reprogram those. But the more common approach here, we're just going to ignore the bottom part here, is to take a single chain antibody. Okay, so you take the variable domains from an antibody that recognize some tumor, and you then clone those or fuse those to the T cell signaling parts. Okay, so essentially what you're doing is making a T cell with an antibody receptor on the surface. Okay, and so now T cells, those T cells can recognize tumor cells like an antibody would, but they still have their killing properties. And so that's really what most of the current chimeric antigen receptors are designed to do. For most of the time, this works well for, again, blood cancers and things where the T cells are going to immediately be interacting with whatever the cancer cells floating in the blood with. Doesn't work as well for solid tumors because you have all these other problems of access and getting into the tumor. But it is a global approach for treating cancers. Now, one of the older approaches, though, is basically going back to alloreactivity. Okay, so we often think of bone marrow transplant as you want an MHC matched donor because then, your cells won't attack your self cells. But in reality, if we go back to allogeneic transplants, if you skin graft the black 6 skin onto a biopsy mouse, that tissue gets rejected. Well, what if we'd use that to target tumors, but and accepting the damage to other organs? Okay, and so typically what this is done here is that you just basically isolate T cells. There's a lot of ways to go about this, but you're isolating donor cells that are not MHC matched. So there's an MHC mismatch. Okay, and so what you then want, you infuse those into the host, and you want them to attack the tumor. That's called the graft versus tumor. Now, typically, when you're transferring allogeneic bone marrow, within the bone marrow is sort of a dirty little secret. There are circulating T cells, and so some of those T cells will actually be ready to respond. And that's where you get this graft versus tumor effect. The downside of that is those T cells can also attack normal organs. This is graft versus host disease. So the graft, here are the T cells contained in that bone marrow, they can attack the tumor, but they can also attack your liver or other cells that are similar to the tumor. Now, what does this rely on? Well, it would be great for tumor-specific antigens, but that's not really allogeneic activity. But what this is designed to do is tumors then lose control of expression of some genes. And so if you have a tumor-associated antigen that's expressed in higher levels in a tumor than it is in your other cells, then you're going to preferentially kill the tumor. And many studies have now shown that if you're doing a bone marrow an allogeneic bone marrow transplant in a cancer patient, if you don't see any graft versus host disease, you're not going to see a graft versus tumor effect. If you see a lot of graft versus host disease, you're going to see a lot of graft versus tumor effects. So they go hand in hand, and it's often difficult to separate the two. Now, one idea has been, okay, we can do vaccination against cancer. And I think this is still a long-term goal. And so the idea is to do first either a prophylactic vaccination, meaning you vaccinate before you ever get cancer. And there is some evidence that this can work, right? One successful example is the HPV-16 vaccine. Has very high efficacy for preventing cervical cancer. And then, and it is one example of a preventative vaccine. But most of the time, you don't have a pathogen associated with the tumor. HPV-16 induced tumors, you can target the viral protein. So you have to make sure that you choose something that's not going to give you an autoimmune response. You can't just vaccinate against a self protein because what if you just induced autoimmunity in everybody you vaccinated? That would be a horrific thing. It has the potential, but you have to choose wisely so you don't induce autoimmunity. The other idea is say, okay, we'll develop a therapeutic vaccine. This means that a vaccine that's only going to be given to a patient that already has cancer. Well, the problem with that is that sort of like is like adding a fuel to the fire. I mean, there already is antigen there. There already is an immune response going on, and so these have shown limited efficacy just because the T cells are already exhausted. They're already expressing inhibitory receptors. You already have set up this tolerogenic environment in the tumor. So therapeutic vaccines have not historically worked because you're just adding more antigen to something where there already is antigen. The way that most clinical strategies are going now is to block the inhibitory receptors that we talked about. Okay, so either blocking, for example, CTLA4, so that prevents the T cells from that would normally prevent T cells from expanding or proliferating from naive to effector cells. There's also blocking CTLA4 on the regulatory cells, so enhancing or blocking their ability to shut down other cells. But that's really saying, okay, we're going to block CTLA4 and get better responses or get more responses, or we can block things like PDL1 or PD1, which are normally going to shut down effector cells. There's a number of these. It's not clear if blocking the ligand or the receptor is better. Both are in clinical use now. But this has become the standard care of approach for melanoma, right? You no longer, it used to be you would do chemotherapy, and if that didn't work, then you would go to something else. This is the frontline approach for treatment of many melanomas, is to go straight to immune checkpoint blockade. But here's the problem, and this is the problem that has been faced in chemotherapy, in radiation therapy, and even in immune therapy, is the idea that cancer, by the time you show up in a clinic with a tumor, that cancer has now been around for 20 years, and there's a billion cells there, but there's a million different clonotypes of cancer within there. So even if your approach kills the bulk of the tumor, there are some of these slowly dividing cells that don't really look like cancers. And these are called cancer stem cells or sometimes cancer progenitor cells. So you're going to kill off the rapidly dividing cells that have high levels of antigen. They're really doing stuff, but then you have these other cells that are just sitting back saying, well, okay, I'm not going to divide. I'm not going to express high levels of antigen. I'm going to sort of look more like a self cell. And so their immune system can't really see them. They're sort of the spies. So this is still mostly a hypothesis. There are a few examples of cancer stem cells, but it's tough to identify them. The immune system has a hard time identifying them, and scientists do as well. And so what the upshot of that is, even if you were able to design an immune therapy that could kill off most of the cancer, these cells are just hiding there and they're not really looking very different. And they may require a totally different approach for killing them.
I had a quick question.
Yes.
Wouldn't the theory be proven if you saw remission in the cancer at a similar location or exact location where a person underwent treatment or had the tumor removed? Wouldn't that be pretty good proof that?
Yeah, but it doesn't happen in all cancers. So for example, cutaneous T-cell lymphoma, which happens in the skin, where you can see the actual tumors under the skin, you can give a therapy that works and those tumors will go away. But when they reoccur in the exact same spots. And so that's some evidence. It doesn't necessarily mean that it's cancer stem cells, because it could be you just didn't kill all the cells, it was not 100% effective, and so there's some leftover residual cells. They don't necessarily have to be cancer stem cells, it's just the therapy was not 100% effective. So that's the problem with arguing that those were stem cells, is saying, well, how do you know it just didn't kill all of them? Which could easily be the case. Okay, so that's where I'm going to end today, and I'm happy to take questions.
Dr. Blattman,
Yes.
for the inflammation as tumor therapy, so does this still work with the cancer types who have already evaded the immune system?
So you're talking about
Where you're just increasing the amount of DAMPs and then T cells to respond to the cancer.
yeah, it definitely can.
Oh,
So the
it's okay.
TNF can cause TNF. We remember is one of the things that will signal for cell death, right? This is what CD4 cells and NK cells will use, is this death receptor. Well, TNF is essentially the death receptor, or it's binding to the death receptor. So it's going to kill tumor cells directly.
Gotcha. Okay, thank you.
Some of the other ones won't quite work as well, because they're, again, you're trying to overcome that suppressive environment, but TNF certainly will work.
When was the idea developed to inject different pathogens into tumors to help cause an immune reaction? Wasn't that developed like a long time ago?
Yeah, that was
Good
that,
idea.
that was Coley's toxin, and there's a whole history of that you can look up. He was sort of given the credit for that. There are some other people who might have been doing something similar, but he was the first one to really implement that as a strategy.
I guess the number, the date on that you presented, that we had evidence that the immune system attacks tumors in 2001, is pretty shocking to me. I mean, I don't understand how that was maybe a possible therapy if it showed remission of some tumors, and then we figure out all these things about T cell receptors and how they're specific for things, and then we don't draw that conclusion until such a later date. It's I don't know. It was just a comment, I guess. I'm just shocked the first evidence of that was in 2001.
Well, remember, immunology is a rather young field, right? Bob Schreiber is still alive. One of my graduate students went to do a postdoc with him 6 years ago. So a lot of these discoveries, toll-like receptors were only discovered within the last 20 years. So yeah, there was a lot we didn't know. The idea of using adjuvant therapy, or Coley's toxin, really is about 100 years old. There are some examples like that you could argue back into the late 1800s. And pretend there's some in history, right, that the Egyptians may have done some of this, but really it's not clear. So some of the anecdotal evidence was, okay, if you dressed a tumor and it got infected, sometimes the tumor would go away. So, but then, around just before Ehrlich, Coley was saying, okay, well, what if we try and do that? Instead of having a live bacteria, though, I'm going to kill the bacteria first and then inject it.
So were they just assuming that there was some bystander effect, and that's how the tumor was being recognized, and that's how they justified it instead of...
That's absolutely right, because they did this. This was done. Coley's toxin, or a version of that, was done all the way up through mid 60s, 70s. I mean, it was a viable approach.
Thanks.
Dr. Botman,
Yes.
will there, or do you, or does another professor teach a cancer immunology course?
Cancer immunology, not specifically. I know that Karen Anderson has taught a vaccine course. Carla Maley has taught a cancer biology course, but I don't think anybody here is teaching cancer immunology, no.
Okay.
Okay, if there's no more questions, then I will see you on Thursday.